For introduction, my name is Iori Yamahata from Nagoya University Graduate School of Medicine. It is a great honor to be here to present our research. Today, I'd like to talk about deep learning-based design of receptor selective cell-penetrating peptide. The intracellular drug delivery is a barrier that must be overcome in order to develop drug design. However, for most macromolecular drugs cannot across cell membrane. To address this issue, cell-penetrating peptide, it called CPP, are gaining attention. CPP is a small peptide, less than 40 residues, and has the ability to improve the membrane permeability of a drug by binding to it. There are two major mechanisms of internalization: direct penetration and endocytosis. However, there are also problems in the clinical application of CPP. The typical examples are low efficiency and poor selectivity. The most previous studies have focused on the efficiency of CPPs, and few have focused on the selectivity. As you know, receptor expression differs from cell to cell. Some cells have a lot of receptor A, while others have receptor B. If CPP can be designed to cause endocytosis only via receptor A, not receptor B, it should be able to deliver only to specific cells. Our method consists of two steps: sequence generation and optimization cycles. I'd like to introduce step 1, sequence generation. The objective in this step is to generate candidate sequences with CPP property. To address this objective, we fine-tuned EVDIF, which is generative model of protein sequence, and using 1,082 non-CPP sequences with LoRa method. The LoRa is a well-known method to fine-tune large model with small dataset while retaining their original weight. After fine-tuned EVDIF, we generated 1 million candidates using fine-tuned model. To check the 1 million sequences generated, they were embedded with protein language model and projected to UMAP. And you can see that the sequence generated by fine-tuned model, shown in blue, and non-CPPs, shown in red, have similar distribution. This means that the generated sequences have the same properties as the non-CPPs. Now, we have 1 million sequences with CPP properties. Next, I'd like to explain step 2, optimization cycles. The objective in this step is to select promising sequences with high selectivity from generated candidates. We repeated optimization cycles 60 times, consisting of 4 sections: docking, MD simulation, training under prediction, and Bayesian optimization. I'd like to explain only the core ideas in order. First, docking. We have to prepare the complex structure of CPP and receptor in order to perform MD simulation. In our case, we obtained 4,000 complex structures by docking 2,000 sequences and 2 receptors. Next, we performed MD simulation to calculate binding energy between CPP candidates and receptor. Performing a 4,000 simulation, we got 4,000 data contained sequence information, receptor information, and binding energy. Using this 4,000 data, we trained 12 deep learning models which can predict binding energy from sequence information. The purpose of training 12 models is to express uncertainty of prediction. Using these models, we predict the binding energies of all candidate sequences. Then we obtained a summary of the binding energy predictions for each receptor. Finally, Bayesian optimization. Based on the prediction summary, we plotted binding energy, like in this figure. If we want to sequence that binds to receptor A, not to receptor B, the upper left point is promising. Then we select this point to be tested in the second cycle. This is the end of one cycle. I'll explain next cycle a little bit. In cycle 2, docking and MD simulation are similar to previous cycle. From 2000 sequence, the 4000 data can finally be obtained. This is an important difference from the previous cycle. A total of 8,000 datasets from both cycle 1 and cycle 2 can be used to train the prediction model in the second cycle. Furthermore, if it is cycle 3, it can be trained using 12,000 data. If it is final cycle, we can use 240 data to train. In other words, the prediction model becomes more accurate as the cycle progresses. A more accurate model allows for more accurate exploration and selection. In other words, by repeating this optimization cycle, promising sequence can be obtained comprehensively and efficiently. And let's move on to experiments and the result. We focused on two receptors, CXCR4 and NRP1, which have been reported to be involved in CPP and cytosis. And we simultaneously optimized the binding ability to maximize CXCR4 and minimize NRP1. And we repeated 60 times. And this slide indicates the result of a prediction model in the final cycle. On the left are results for CXCR4, on the right are NRP1. Comparing the upper and the lower result shows that the prediction model captures the overall trend of actual data. This is a result of Bayesian optimization. The left figure indicates a feature of the sequence selected by Bayesian optimization in each cycle, showing that we were able to sample from specific lesions. And the figure on the right indicates the binding ability calculated by MD simulation. In our experiments, the goal is to maximize binding ability to CXCR4 and minimize to NRP1. So the upper left point is the desired sequences. We can see the updated with the most promising sequences shown in red. And finally, we were able to obtain 9 promising sequences indicated by red dots. In this study, we developed a combined deep learning and simulation method for receptor selective CPP design. And we successfully optimized and obtained high-quality candidates without the need of exhaustive testing. And we plan to validate our computational method. Also, limitation. This study did not consider endosomal escape. And to further improve peptide selectivity, it is necessary to integrate the studies focusing on direct penetration. Although challenges remain, we believe that this research can contribute to the development of next generation peptide. Thank you for your attention.
So any questions from the audience? Okay, so I have a question. So you explained the current limitation of the current approaches. And what is your idea to improve to improve the method so that this method can be applied for the currently not suitable problems? And could you give me a detailed idea about your next plan?
Okay. So I said this study did not consider endosomal escape. This endosomal escape is necessary phase of internalization of CPP. Indicate this figure. So we have to consider the penetrating to this endosomal membrane. So therefore, we have to simulation or some calculation to improve this phase. Does that make sense? Thank you very much.
And please.
Thank you very much for your interesting talk. I have one question. To predict the binding energy, you constructed the 12 predictive models. What is the difference of the objectives between them?
Okay.
I might have missed your explanation.
Okay, thank you very much. You're referring this slide? Okay. So major objective is to explain the uncertainty of prediction. So we trained the 12 model, but this 12 model has different architecture. So each model can run their sorry, confusing. Okay, so okay, can I explain after this session? I'm sorry.
Discussing the break.
Yeah, sorry. Thank you very much.